Cool, so I think we're going to start again. We're now heading into the color domain. So the color domain, I have briefly hinted at a few times, but now I just want to check that you understand what I've been saying. So can digital cameras, or what you have in your microscope, whatever, can these things actually see color? Okay, so what do they actually see? They see they see varying intensities over some sensor that you have now defined as a matrix of values. So they don't see color as much as we think they see color. It's like, look at all of this amazing color that my image can see. Now, what you have to also just remember what's happening here is this is a multi-stage thing that's actually going on. You first of all taking these intensities of light, you discretize them, is that the word? To make a grid of values from them. And then this grid of values is not helpful for us to visualize, so we just make our computers and our screens in such a way that we can then represent these matrices of values again in a way that is appealing to our eyes. So screens are designed for our eyes to somehow understand what's going on in the data behind the screen inside the computer, but it's not really the data. It's not actually color. It's not actually an image. Okay, so this is now a CCD sensor. This is a little bit old technology. Most modern sensors use CMOS, but the same principle applies. So how the sensor is composed is you have all of these light-sensitive elements with a little filter in front of them. So they essentially filter out different wavelengths of light which you don't want, and only the wavelength of light that you do want it captures, but it makes it very specific. So it's only capturing exactly at the green, exactly at the red, or exactly at the blue. Or pretty exactly. There's obviously always some fall off there, and because of the different, how can I say this? You need to put them out in some kind of a grid form. So there are different layouts of these photosensitive elements that are designed in a sensor, but they're never actually exactly on top of each other. They're always kind of put out as some grid, and then this little grid is then what is associated to a single pixel. So you can now see for this one. Let me just try to get the laser pointer. For this one, there are actually 4 cells there, and then these 4 cells then produce a red, a green, and a blue component, which is then assigned to a single pixel. So just in terms of spatial resolution, there is actually a little bit of additional loss. Your sensor is a little bit more sensitive, but it loses a little bit of the spatialness to it because of this very slight nuance of not being exactly at the same location. But then it also only captures very discrete wavelengths of light. It's not the full spectrum. Now, obviously, the full spectrum looks, let's say, for example, it might look something like this. The thing that is actually going on in the real world, the photons that's actually reaching us, have a certain amount of energy in these different frequencies, for example. And then the sensor needs to somehow translate that to something that's discrete. And as I was saying, these photodetectors are very sensitive to a specific frequency, and they have a little bit of a fall off there. But it's going to be some combination of these 3 that they end up capturing. So if I want to capture this entire spectrum there, I end up capturing some portion in the red, which corresponds to these range of wavelengths. I capture some portion in the green, which corresponds to these spread, and then the red also in the latter part. But what this kind of implies is that I can't actually ever get the true representation of the signal out, because all of this entire intensity where this photo element is sensitive is going to be combined into a single value, 205 intensity. But it's actually corresponding to intensities in this entire range of wavelengths. The same thing for the green, same thing for the red. So there is a lot of loss of information already happening in the capturing because it's trying to give a discrete value to actually a spectrum of values, a spectrum of intensities. That is unfortunately the best that we have in our typical microscopes, but it's just good to be aware of that. Don't read too much into the pixel value intensities themselves, because it actually corresponds to a bit more of a range and an overlapping range. So you capture now in something that is supposed to be blue, but then you capture some green intensity as well. Your green sensor picks up something because it's actually sensitive in that region. And then it says, hey, there's some green signal coming out, but actually it's just blue. It's just that it captured in the same overlapping region. Okay, so why are we using red, green, and blue? Now, the first benefit is that you can see that we can represent almost any color you can think of with this red, green, and blue spectrum that we defined. And it's kind of based on how our human eye perceives light, because the context in which cameras were developed is we want to capture a scene so that we can ultimately represent it in some way that our eyes can appreciate and think this is a good photo. So it's all based on human perception, not necessarily on what is most helpful for biological analysis. And human perception primarily here in the context of I want to take photos of you guys, not take photos of cells. So our eyes, you might all of you be aware of this, but I just want to emphasize that our eyes have different light sensitive cones in our retina, and those that correspond to color. There's one cone that corresponds very strongly to the blue spectrum, and then you've got two cones which are somewhat overlapping for the green. One is more sensitive to the green, other one is more sensitive to the red. And usually what happens in people that are colorblind, for example, is these two just shift a little bit too close to each other, and you can no longer distinguish between red and green. And if someone is not colorblind, there's enough separation between the M and the L cones so that you can pick up a little bit more intensity in the one cone than the other one, and then your brain just interprets that as some continuous color intensity. Now, as I was saying, that's the exact same thing that we then use in our sensors. We kind of model them on how our retina sees light. We have these different cells which have different quantum efficiencies over different regions, and then they just capture intensity, and then we have the brain, which is now the computer CPU, that then processes this in some way to give us some visualization back on the screen, which is helpful for us to visualize. But if I have an RGB image, behind this, there would typically be 3 channels. These 3 channels, as I've been emphasizing, does not correspond purely with this is the only red component or only green component. There is some overlap between them, but the point is once you combine them, then they are visually appealing. Now, what happens with these different channels? What do they capture? So white would be in all 3 channels. So white light is basically a combination of red, green, and blue. It appears to your eye as white. If it's yellow, you would have red and green, but nothing in blue. It's basically black there. And then something that is, let's say red, you would have this red flower clearly showing, but in the green component, it actually looks dark. So different aspects or colors have different intensities within these different components, and then you perceive it as a color image if you combine them. And just to emphasize the point of the matrix of values, and that there is no such thing as an image really, it's just a lot of matrix of values that are then given some color associated with that thing that we want to look at. But behind the scenes, there are just these values sitting behind every single pixel. Okay, now I'm going to quickly talk about different color formats. So our sensor captures it in RGB, but we might not want to store it in RGB, or we might not want to do analysis in the red, green, and blue components. We might want to translate this to another color space or color format so that the analysis is helped, and I'll try to make some examples here of where this helps. RGB, the reason why this is the most common format is because our camera captures it in RGB, so that kind of is the default. It makes sense. It's also very helpful for programming. Generally, when you do programming for a computer, you want to render something to the screen. It's helpful to think I'm going to render this color to the screen, which has red, green, and blue pixels on the screen. So it's very, very common to think of it in that sense. But it doesn't correspond proportionally to our perception of colors. I made that point for the grayscale. It's the same thing for red, green, and blue. If the intensity is very linearly, our perception is not linear of that change. Just as a silly example here, if I take a red pixel that's 100 intensity of 100, and the green and the blue, I believe I made 0 here, if I remember correctly, then if I make it 200, this is a doubling of the value. But would you say that that dot is twice as red as that dot? How do you even actually quantify twice as red? Like, what is the perception of it? But the point is that it doesn't really, the RGB format doesn't correspond with our perception of color. And basically it's just a combination of these 3 that then end up making a color. And if I remove some red, add some green, add some blue, then you can change from one color to another. But then again, minus some red, adding some green and blue, doesn't intuitively tell me that I'm going to move from this to that. That doesn't translate with my intuition. So that's why we have some different color spaces or color models, or it's got different names. One of the other ones is the HSB, which stands for hue, saturation, brightness. There's a basically similar nuanced thing in a different definition, but it's called HSV sometimes, where the brightness is just substituted for value. And what you do here is, instead of having 3 orthogonal axes for red, green, and blue, and then combining them to create some color, you now split it up as a radial circle basically for the hue, where you say hues are going to change along a radial path. So I can say if I'm at purple and I subtract some hue, then I'm going to get to, oh no, this is adding, sorry. If I add some hue to that, I'll end up with red. If I add some hue to that, I'm going to end up at yellow. That's a lot more intuitive. Like we have the rainbow ingrained in our understanding. So if I just add to the hue, then I just naturally move along the rainbow colors. Okay, so that the color is intuitive. Now saturation just means basically what is the brightness or the liveliness of the color. So if it's full saturation, you have all of these very vivid rainbow colors. If it's low saturation, everything becomes a lot more muted and dim and goes to more grayscale. So again, saturation has an intuitive understanding to it. Value is then basically how bright it is. So high value is its full brightness, low value is its black, basically. So again, that is an intuitive understanding. So with 3 intuitive parameters that I can, with my visual perception is linear, it's my intuition behind it is easy to understand. I can easily move from one color to another. The HSB is quite a helpful alternative color map. I basically said all of this already. Okay, then there's another one which is called, I don't actually know how to pronounce this. Does anyone know how to pronounce this word? CLab. Most people just refer to it as lab, and it stands for the L and the A and the B components. I don't really know why it's called A and B, but anyways, the L stands for lightness, where lightness is a value between 0 and 100, very similar to the value that we had for HSB. And then we've got 2 orthogonal axes, and the A axis corresponds to the green-red axis. So you can see there, maybe this is better. If I move along this A axis, I transition from green all the way to red in a continuous way. And the B axis corresponds to a blue-yellow axis where I can change from the one to the other. What's interesting is this is still very intuitive in reality, even though it might not seem like that, because if I basically have this disk, if you can think of it like that, that's made up of these 2 axes. And then you can think of it almost as Cartesian coordinates that I'm moving around on this disk. And then it creates a nuance of both the color as well as the what do you call it, saturation in a sense of how vivid the color is based on how I move along this grid. So it's still quite intuitive. And then the light is obviously just the brightness of it. I just want to do a quick demonstration of this. So if I just take this, you don't need to follow along, this is just so you can see how it works. I'm just going to take this example, this Nile bend image. Maybe I must just again try to make the screen black. Okay, there's my Nile bend. So this is a color image, RGB image. By default, it's RGB. So if you look here under image type, you will see that it's an RGB color. I'm just going to maybe also show you I can duplicate an image. Then I've got another copy of the same one. I could, for example, make this an 8-bit image by simply, let's use the mouse. Clicking on 8-bit, then it's making it, instead of 3 channels, red, green, blue, now it's just combining it into 1 channel, which is now a grayscale image. I can also change this to HSB stack or a lab stack. I just want to show you those 2. Let's start with RGB stack. I think that's probably a good place. So this is red, green, blue. Now it's not currently showing it as red or green or blue. I can probably change that with lookup tables. Red, I'm not sure. I probably first need to separate the channels. Let's just quickly try it. I'm a little bit winging this one. I'm just going to do color split channels. Come on, okay, now it's I still have to do the lookup table thing. This is blue, this is green, and this is red. So these are the three components. It gives some information, but for what I might want to do with it, it's not necessarily extremely helpful. So let me just show you the other 2, which is, I think, a lot more intuitive. Open sample, Nile bend, and I must download it again. Okay, duplicate so that I don't destroy it again. If I now go to type and I make it a HSB stack, now this is hue, saturation, and brightness. So you can see brightness kind of obviously corresponds to how bright the region is. Saturation is how vivid the colors are. Well, all of the colors are somewhat muted, so they're not very vivid rainbowy color, so you can kind of see that here. And then for the hue, so you can see the hue here is similar, and then the hue around this part is also similar, so the color associated to it. Just pay attention to the river. So I can maybe see the river in this one somewhat clearly, where I'm looking at the saturation, because the river is pretty black there, so the saturation is pretty low, but it's not as clear. So now I'm going to convert this same image to a lab stack. And, okay, I actually converted the image. But now I've got the lightness. So lightness, again, similar to value before, it makes sense. And then the A component is that, what was it now, red? I forgot now the two colors. It's the green-red axis, and then the B component is the blue-yellow axis. And you can see now that I have the blue-yellow axis, I can actually see this river pretty clearly. So if I wanted to, for example, just extract the river, going to this lab and then using the B component is the easiest way to immediately get that without needing to do any other processing. I just, looking at it at in a different color space, I suddenly get more information out of the image. So being aware of different color spaces for different problems is just good to consider. Okay, now I just want to make a comment quickly on file format and why it matters. So how you store your images does actually make a big difference on the data that is stored in these images and what you can ultimately do in analysis. So let's just quickly think about what the differences are between the common formats like TIFF, PNG, JPEG, and GIF or GIF. I'm going to call it a GIF. What is compression and how exactly does it do to the image? So what most image formats try to do, except for something like bitmap, which it's got zero compression, where you just basically have your bitmap of RGB values just stored in as crude as way as possible, and then you just open it directly like that. All the other formats try to at least compress the data in some meaningful way. Now, what compression is supposed to do is you have your original image, which if it was just the bitmap, it would have taken up a lot of space. After compression, it takes up a little bit less space. Obviously, we like it to take up less space, but we don't want information to be lost. So in a lossless compression format, we can make it smaller. It actually takes up less space, but then when we restore the image or uncompress it, then we still have our perfectly identical image to our original image. The other way is that we could compress it in such a way, we might even get a smaller size, but then when we restore it, we can never get back to the original intensities that we had. And there might be some artifacts that are introduced, some undesirable things, and that means that our analysis done on that lossy image now is not going to be as accurate. And if you want to make statistical conclusions, on what is your stats now based? On a muddy, messy image. So very important to consider. Just maybe some quick comparison. I'm not going to go through all of the details here, but you can look at it yourself when I share the slides. But JPEG is not a good format for bioimaging. Just in general, stay away from JPEG, don't use JPEG. I'll show some reason why just now, but it's a very lossy compression. The goal of JPEG is to make images kind of as small as possible so that they can be transferred over the internet and things like that. And you just want to get a good representation of your image to the other side. You don't care about the perfect representation. It also doesn't support Z-stacks if you work in that domain, like confocal, for example. It also doesn't support higher bit depths, so you're kind of stuck with 8-bit images. 8-bit now for red, green, and blue, so that's why it says 24-bit in total. Okay, PNG, this is a lossless compression, so you don't throw away any information. It allows transparency if that was important, but not so much in bioimaging. It doesn't support Z-stacks as well, but it does support higher bit depth, so you can always work with the higher resolution images there. TIFF, this is a lossless compression. This is probably the most common compression that you will see in any bioimaging because it's a universal format. It's not tied to a specific brand or anything. It's universal. It primarily supports Z-stacks, so we can have stacks in depth, and we can have stacks in time. It contains a lot of metadata, so there's a lot of great things about the TIFF format, and that's why it's primarily used for that. It also supports higher bit depths. Okay, now just a mini rant on JPEG. I'm not going to go into too much detail, but if I have my original image and then I just, for JPEG you can decide what compression value you want. And if you make the compression much higher, or actually technically in this case a lower compression value, then the size of the image becomes smaller. So you can see nicely it started with a 75 kilobyte image and I end up with a 5 kilobyte image. That's quite a substantial gain in terms of space. But in the process, you started with nice clean edges, and now you've got all of these muddy edges going on here, and if you were to plot it three-dimensionally, you can even see it more clearly that there are these ripples that happen. And these ripples almost always happen at the edge transitions, or in the what you call high frequency regions. So what is a high frequency region? When you're quite flat, very low changes over a long period of time, but when you have a sudden drop, that's a very high frequency region, and JPEG doesn't do well with that. It basically tries to get rid of as much high frequency information as possible, and therefore to get rid of that, you have to blend the edges a lot more in order to compensate for it. Okay, and you probably have seen the JPEG artifacts. I don't know how clear it is here, but like for example, that muddiness, if you see that in an image, that's just JPEG compression, not using a great compression ratio. All right, so Fiji. One of the great things about Fiji is it's a generic tool. So many of the other pieces of software like Zen and Zen Black and all of these things that's tied to a specific brand of microscope, they can do a lot of analysis and everything, but they are tied to that file format of that brand that you're using. Fiji solves this problem by introducing this idea of bioformats, which can work with multi-channels, multi-time, in the multiple focal planes. So you can even have lots of positions that you set up. So I can have positions, I can have time-lapse, I can have Z-stack, everything in whatever format that was captured with all of the metadata. I have now combined in one package in Fiji to be able to deal with all of that type of images. So for example, Zeiss would have the CZI format, Nikon would have the ND2, I don't know how to say this, Leica, how do you say it? Leica, thank you. Leica is a LIF format and Olympus is an OIB format. And all of these formats have some similarities, but they all are too different and you can't just work with them interchangeably. So that's where bioformats come in and it solves that problem. So bioformats is a plugin in Fiji which basically supports 150 file formats. You can look at all of the detail here as to I don't know what, whether it supports pixels and metadata for these different formats, but for most of what you would care about, it supports that out of the box. Okay, now a big thing, as I've been emphasizing, is this metadata. So when we have the image, it's not just the image information that is important to us. We also want to know under what conditions was this image acquired. We want to know what is the resolution of the image. If we want to put a scale bar or something, we need to know how many pixels corresponds to how many micrometers. So all of these things are contained in metadata, and if you, for example, don't save images properly, you often throw away this metadata, which makes analysis later on very challenging because then you have to either guess or remember, and then it doesn't work. So keeping the original files is usually preferred because then you always have access to that metadata. And then you can always just import it into this bioformats thing and it will automatically just read the metadata for you. This is what it might look like. It's usually in an XML format, the metadata that it's stored. So it's all of these tags and then with subtags and subtags and subtags. So it's kind of difficult often to get to the thing that you're looking for. And I'll show you how Fiji deals with this, which is not necessarily very elegant, but yeah, I'll get to that just now. So when I was busy transitioning in this presentation to focus not so much just on the raw pixel data, what is sitting behind our images, what is the, where did these pixels come from, like looking at lookup tables, color, and all of these things, that was actually just focusing on the raw pixels. Now we're going to get a little bit more into visualization, how do we enhance our images so that we can get more information out of it, and ultimately, how do we do segmentation. Okay, so I think I should have done that part that I wanted to do here. So let me just, it seems like I'm heading into some other piece now. What I wanted to show you is if I import an image, you don't have this image with you, but where did I? Okay, so I've got this image here. This is a .nd2 image that was captured with a Nikon microscope. I'm going to click here. This is now the bioformats plugin that I was talking about. When you try to open an image with bioformats, it asks you for a lot of parameters. Now, you can do a lot of things quite easily here. I can, for example, the image I'm currently importing is both a Z-stack and a time-lapse sequence. It's only a single channel, though. But I can already say, as you import it, split the channels for me so that all the channels, the red, green, and blue, whatever might be separated out, that I can see them as separate windows in Fiji. I can also say split out the time points, that every single time point with its Z stack would be a separate window, and I can maybe find that helpful. I can, if there's a series of different positions, I can say open all of them in a series. I can use a virtual stack where it doesn't read the entire image into memory all at once. It keeps most of it on the hard drive and then it just reads what it needs whenever it needs it. So that's sometimes helpful if you work with very large images. And yeah, then this display metadata. So usually that's ticked off. I'm just going to turn it on. You can also change the color mode if it's a composite or color or grayscale or whatever. So there are some parameters here to play around with, but let's just import this image. It's going to take a little bit of time, but yeah, you can see the metadata so long. So this is how Fiji represents the metadata for this image. It just has a key on the left-hand side. I'll look at the image just now, and then it has the value for that key on the right-hand side. But it's just a very long list of things. Now, a lot of this is not useful at all for what I would need for analysis. So it's a little bit cumbersome to work with this. You can, I think, find, and then I can say, for example, what is the wavelength, and then I can get to a wavelength pretty quickly. But I find it usually a bit easier to just say save as, and then you save it as a CSV file, for example, and then I can open it in Excel or something, and it just makes it a little bit easier to then color things in and find values or something. But this is up to your personal preference how you want to work with this. But yeah, I can set up some filters in Excel pretty easily to get rid of some of the fields I don't care about. And we're going to look a lot more detail at this metadata when we get to deconvolution. I'm just showing you for the moment that you can get it out. It's in your images. It tells you everything you need to know about the image. So this is the image that I imported. You'll see that I can't see anything. It's too dark. So let's do that thing that we did before. Let's just turn it to an auto. Turn that up a little bit. So this image that I just imported, it's mitochondria. You can see that's the Z stack. And then I have, on another axis, I've got time. And quite importantly, everything is in the context of what it was imported. So I can see that this, it's quite small, but I can see this is a 16-bit image, for example. I can see the resolution of this image is 1200 by 1200. I can see where I am in the Z stack. I can also see what microns it is. It's 132 microns by 132 microns. So I now know what each pixel would be in terms of microns. I can then do certain things such as use that information to, I think it's here under analysis tools, and then just put a scale bar on it. This is a very simple thing. Where did it? Should have added the scale bar. Ah, there it is. So they just added the scale bar. Now it was only able to add that scale bar correctly because the metadata was on the image. If you were to save the image in many ways and you've destroyed the metadata in the process, it is kind of impossible to get back to what is the scale of a single pixel. Now, usually when you do analysis, it is very important to know what is the actual volume of something in micrometers or length of something in micrometers, because we're working in a biological context where things matter, like size matters. So metadata, very important. Always try to keep the files in the original format, and then just you can always save it and then visualize it in whatever way you want, but always keep the original files if you need to get back to that metadata. Okay, so now I am finally getting onto the more visualization things. So this is the next, or this is technically the first image in that image pack that I gave you. So we can maybe just all open up Fiji again and then get the first image. It should be the folder listed 1 underscore something. It's called single image. Just want to check everybody's got it open. All right. I forgot to mention this, but if the metadata is available for an image, then you can click on image and then show info. Then it will show you, it will pop up this window and then show you the metadata as well. So if you imported the image without asking for the metadata, then you can always get it later. This image, you will see that it doesn't really have that much metadata. It's got some information here. It's not nothing, but I don't see scaling, for example. Yeah, it tells me it was captured with the Olympus objective and things like that. But yeah, I don't see any. Yeah, the micrometer scaling aspects have been thrown away in this image. But okay, so this is our image. Let's just look at a couple of things around it. Again, this is a 16-bit image. So if I was to then look at intensities, and I want to just show you a quick way of how you can find intensity at a specific point. If you just hover the mouse over that point, you can see on the top there, you see the XY coordinate, and then you see the value that's actually happening there. Now, one way that I can know that this is 16-bit images as well is that the value is much larger than 255. So there's a lot of nuance in the detail, in the intensity there. I can also visualize the histogram, Control H, there's the histogram. The histogram is clearly, everything is biased towards the lower end because there is so much black in this image. So again, it's very helpful to click on this log, then I can just see more detail as to what's actually going on. There is actually intensities all the way almost to the very top. It's not saturating. If it was saturating, you would have a peak here at the top. Let me just maybe show what that might look like if I'm just going to click here enhance contrast. I'm just going to make it saturate. Let's make it saturate with 5%. This is now massively skewing things. Did it not actually do it? No, it didn't actually. Oh, I think I was on the wrong thing. Okay, this is image, enhance contrast. And just make it saturate with 5%. There, okay, now it's nice and bright. If I now look at my histogram again, okay, it's not very clear, but there is some saturation that's going on there. Let's maybe saturate it even more. Process, enhance contrast, saturated, I don't know, let's just make it 10%. Okay, things are getting nice and bright now. Log, I don't know why it's not changing. There should be a whole lot more saturation than that. Was it not saved, maybe? Let me just check here. Okay, apply. I'm going to click apply here. Okay, now it should be correct. Now if you look at the histogram, there is actually a pretty big peak of saturation there. If I view it in the log domain, you can see everything is now shifted up, but there is now saturation there. So you want to always, as I've been saying, try to retain as much information as possible. So saturation is not a preferred thing to do. So let's see if there's anything else. Okay, that's it for that image. We're going to use this image quite a bit later on. Maybe just to show one thing as well, which is always helpful, is just making a selection and then cropping that out. So if I want to get these cells by themselves, I can just select this by clicking on this rectangle tool. I'll select the cells, and then I can go image, crop, and then it separates them out, and then I can do whatever analysis I want to do further only on that region. You can also I don't know, view the histogram of that region or whatever I want to do. So it's yeah, helpful to just in general be able to do that.
On the cropping tool,
yeah.
Can you set, I want a 200 by 200 crop, or do you have to manually always crop out like?
Yes. Let me just show you. I'm not sure in the software. I usually do that with a macro. But let me just check. Maybe there is a way. If you click on this little arrow, it's usually gives you more options. Come on, there you go. Stroke. Yeah, I'm not sure that it allows you. So let me just show you how I would do that to answer your exact question. I would say macro and then record macro. So this is on the plugins, macros, record macro. And then there's this window that pops up that's going to keep track of what am I doing, but in a code type of way. So now I've made that selection, and then I can create this macro, which is just the code there. I don't know how to increase the font. Let's try to. Okay, so this is a very simple function, but now this is the x and y starting point, and then the width. I think this is the width, so I can make this, for example, then 200 by 200, and run this macro. There's the run button. There it appears again. And there it has a perfectly square crop. So that's generally how I would do it. There might be a way to do it graphically speaking. Like for example, I think if you hold shift while you're dragging it, then it retains the aspect ratio. But I don't know how you start. I don't know, if you hold control, it does this. Oh, okay. If you hold shift, then it keeps it square. Okay, so it doesn't matter how you draw it. If you hold shift, it keeps it square. If you hold control, it does it. It enlarges it around the current position. Alt doesn't do anything. So yeah, that's keyboard shortcuts. They're helpful. Okay, so that's it for now for this image. Like I said, we'll get back to this image just now. Just want to get quickly to my presentation, check that there's not anything I missed saying here. Yeah, maybe just a comment on this. So the bit depth of this image was 16 bits, but if you look at the actual range, it doesn't go to the full possible range of 16 bits. So it will always show you the maximum value that is actually present in your image. So if this value is surprising to you, it's just because that is the maximum value that is actually present instead of a true representation of the available histogram if you were to use all 16-bit bit depth. Yeah.
How do you know then that it was a 16-bit and not something like a 12-bit, because we said 12-bit is 4,000, so we wouldn't know if it's...
Yeah, so actually a lot of microscopes have this tendency to you capture in 16-bit, but in reality there's only 12 bits of usable information. Let me just show you how I know that this is a 16-bit image. If I open the image, at the top it shows there 16-bit. So that's, according to Fiji, what it thinks the image resolution is. Also, if I go to image type, then it will show, come on, type. I can't get the mouse there. Let's use the actual mouse. It shows 16-bit there. Usually in show info, it should also say 16-bit here somewhere. There, it says bits per pixel is 16 bits unsigned. But often there it will then say that the microscope limited to actually 12 bits. So it's 16 bits of image storage that's used, but only 12 bits is actually information.
Isn't that a waste of space?
A little bit, yeah. But it's just generally why that is most likely, unless if you've got a very special file format, is that everything in computers work with groups of 8 bits. So 8 bits is called a byte. So you would then have 2 bytes per pixel, but then it can't store it with like 1.5 bytes necessarily, so that's probably why. Okay, so the next image to open up is this one. So if you go now to that, I think it's the second folder, then you can open up these separate images. Again, it's probably easier to just select them and drag them across. Okay, so I've got 3 images here. They were stained with 3 different dyes. I'm not going to be an expert in the dyes themselves. Lisa would know a lot more, but the DAPI or the API, that corresponds with the nuclei. Then this CY5, not really exactly sure what that is. Oh, okay, so it's obviously binding to some structures within the nucleus. So, okay, and then this dye is GFP, so green fluorescence protein. Not exactly sure what it bound to. Might be the cytoskeleton. Not exactly sure. But anyways, so you've got these 3 images now. Imagine now you acquired this on your microscope, and you set up, you stained it with these 3 different probes, acquired it properly. Great. Now you, as maybe the image analyst, just get these 3 images, or it might be even in a single stack where it's not just different channels. But long story short, you've got these separate images. Now, for visualization purposes, again, it's helpful to sometimes just assign a color to them. So it might be that, okay, this is the green fluorescence protein, so this should be green. Come on, there it goes. This one is the nucleus dye. That one, you maybe know the dye's emission spectrum is in the blue range, so let's just make this one blue. And then this one is in the red range that it emitted, so let's just make it red. So this is roughly what actually happened under the microscope. If you were to look at the sample under the microscope, this is roughly what you would have seen. This is not exactly, this is obviously assigned colors to the color map, but it gives you a good idea so that you just keep your bearings. Now you can look at them individually like this if you want to. You can also combine them into a single image by going to image, then color, and then merge channels. Just want to make sure everybody gets to that menu item. First of all, it's image, color, and then merge channels. And now you can select which of the images that are currently open. It's going to show you all of the currently opened images should be assigned to the red channel. As I was saying, it's the CY5 in this case. The green channel should be the GFP one, and the blue channel should be the DAPI one. And then you have some options. You can ignore the source lookup tables or whatever. You can not ignore them. Create a composite image, create an RGB image. You have that option here, and then keep the source images, or you can remove them if you don't want to see them anymore. For now, I'm just going to keep the source images. Just create a composite image. A composite image is now it closed it. I forgot to tick keep source images. Anyways, so now you see a single image. You've got this slider that seemingly does nothing, but if you look at the top here, you'll see that it's currently the red channel is selected, the green and the blue, so it actually changes this, and when you do analysis, it will affect that thing, that channel that is selected. So let's just make, I don't know, this one yellow, for example. So since I've now selected initially it was the red channel, now I've made it yellow, but it's only that channel that has changed because that was the channel that was selected. Let's just make that channel red again. Now it's red again. And so you still have access to the individual images, but it's just showing you nicely overlapping so that it's just easier to understand the context of everything. Now you can obviously zoom in and then see, oh, there's the nucleus with the little dots inside, whatever they might represent. Not a biologist. Yeah, then you can split it again, or just maybe before I split it, just want to show you that if you look at the image type, it still shows 16-bit. It's not an RGB image. It stores 3 separate 16-bit images, which is just shown in a composite view, but it's not an actual RGB image. If you were to make it an RGB image, you can click on RGB color there, then it will actually create a single RGB image, which I can't change the channels anymore, and anything I do now will actually change all of the channels. So if I adjust brightness contrast, it will change this for the entire image. So it's no longer separate, where if I had it as separate colors, separate channels, where is it now in my composite image, like here, I'm just going to maybe crop this out. This is going to be kind of a nice little region to look at. So if I've got this one, which still has the separate red, green, and blue, if you look at this adjust brightness contrast now, you'll notice that if it's selected on the red channel, it shows that this is the red histogram. This is only the red channel's histogram. If it's on the blue, this is only the blue channel's histogram, green channel, and then the blue is it's only that channel. And I can then change these histograms independently. It changes only that channel information because remember, it's still 3 separate images, but it's nicely showing it in a composite view. And as I was just making the point, if I was to then change this to an RGB image, then it will actually fix in those parameters. So whatever I changed here is now locked in, actually saved as the actual pixels here, and I no longer have access to the individual color channels. I just work with this as a global individual image. Okay, it doesn't matter whether you have a composite image or an RGB image. You can always separate the channels again by going to image, and then color, and then split channels. If you wanted to check that, image, color, split channels, and then it splits them up again in the red, green, and blue components, which we're back to where we started, basically. Okay, any questions on color images and how to work with them, splitting channels, merging channels? When is it actually one image? When is it store the separate ones, just composite? Okay, everybody seems happy. Let's go back to the slides. Okay, so I just kind of make the point here. Maybe just the last comment here that the RGB you'll see at the top there, whether it's an actual RGB image, which is a single image, or whether the channels are still separate, if it shows 16-bit at the top there. Okay, so importing, we've been looking at importing a single image. Now, I'm just going to demonstrate importing a sequence of images because, okay, let's say it like this. When you capture an image from a microscope, usually it's in a single file. It might be a .czi file, which contains your nice time-lapse sequence and everything. But it could also be that the way that it was acquired, it was actually stored as separate individual images. And then how do we don't want them to be separate individual images. We want to have them one unified thing in Fiji. Now, the way that you would do this is you go to, and you don't have these images, but I'll just demonstrate this. Let's just close everything, that it's a fresh start. There you go. So you go to File, where you would have typically just been able to say Open and then select an image. Now you can go to Import, and you actually have lots of options here, but the one we're interested in is Image Sequence. And if we say we import an image sequence, then I must select where the source of this image sequence is. I'm just going to take this one. I must just remember how it's actually stored. Oh, come on, it's there. So just to show you this is currently RGB images. These are actually JPEGs. This is a terrible example in that sense. But I can import this image sequence and then first of all just identify if there's any unique way I want to separate it out, or is there any way that I want to sort it? Is there an order to them? So then I can say in this folder is my image sequence. This is not image set, just that one. And then it says, oh, I picked up 14 images here. Is there some filter that you want to apply when you import it? Is there some sorting that you want to apply, for example, sorting them by name? And I can click OK, and then great, now I've got a unified thing in Fiji that I can easily just handle however I want to. To show you another example here, if I've got another image set, this one contains red and green. So there are some of the channel is green, some of the channels are red, so I want to now import them separately. So I can say then import image sequence, select my image set 2, that's the folder in which all my images are lying. It says, oh, it picked up 50 images. Do I want to just import all 50? Well, no, I don't. I just actually want the green ones, for example. So then I say, okay, well, all of my file names that are green contain the word green, so I just put that in as a filter. Okay, now it says it's picked up to 25 images like that. And then I just open that. It takes a little bit of time.
So that's basically an inclusion filter. Everything with that in you open.
Yes, exactly, yes. That's an inclusion filter, yes. So so then now here's my green channel. Okay, now I want to do the same thing for the red channel, so I can then say file, import image sequence, and then I want everything called red with red in its name. And then sort them also numerically. Okay, so now I've got my red stack here. It's a little bit unclear. Let's just make it a little bit brighter. That's maybe a little bit too much. Something like that. Okay, so, didn't do it for the stack now. Ah, that's irritating. I'm not going to bug with this too much now. The thing I just want to show you as the last step is then I can do that thing that I was just showing you, where I can say color, merge channels. The red channel is, they've got the same name. That's not very helpful. Let's just rename this window. This one I'm going to call, edit. Where is it now? Rename, image rename. So let's just call this red, and then I can at least pick it up separately. Color, merge channels. The red one is red, the green one is the other one, and create a composite image. Oh, sorry, I think I messed up a little bit here. Sorry guys, these were actually RGB images. I just have to repeat that process quickly. So let's just do the green. Yeah, see, it's actually RGB. So before I do all of that composite things, I first have to just turn this into a grayscale image. Let's just use 8-bit. And I'm going to rename this immediately to red. File, come on. Import image sequence. Green. I think I did initially import the green one. Sorry guys. Okay, I have to now do the red. File, import, same thing but red. Okay, change this one also to 8-bit. Rename this one to be red. The other one to green. Image rename. Okay, we're nearly there. image, color, merge channels. The red and the green, it automatically picks it up from the names. And hopefully this is now correct. Come on. There you go. So there's the two overlaid. It's a little bit dim. Let's try to enhance the brightness a little bit for the red and then the green channel. The red is still very dim, but let's just do the red. This one. There you can start to see the red. Okay, great. Sorry, that took much longer than I thought it would, but you get the idea of working with an image sequence. You can make it a Z stack and then combine everything still in a whatever way you want to do the analysis. Right. Yeah, so this one, I think this is actually a little animation. There you go. You can see that this was now separate images taken over some period of time, and then now you have it in a single stack, so now I can actually see the animation play off without having to work with separate images. Right. Just the last thing, I'm not going to demonstrate this, but you can also, if you've got two different experiments, you've got this wound healing thing where you now have a treated group and an untreated group, and you've got two time-lapse sequences, and you want to compare them to each other. There's a way to go to image stack tools and then combine. Then you can just say, okay, I want to combine them, let's say vertically, and you just put them on top of each other. Then it's easy to exactly compare these rates just visually. Again, this is a visual tool. This is maybe also a communication tool. You wouldn't necessarily use this for analysis because when you now analyze this, this is now considered one image, where it should definitely be two. But yeah, for demonstration or visualization purposes, definitely a very helpful tool. Okay, I did show the image sequence idea already, where you've got lots of images that you can combine into a single thing. Now I want to talk about Z projection. So we've now, you can either import your image directly from your microscope, then you've got your Z stack images directly, or you could create them from this image stack, but you've got a Z stack. That's not the big point here. The more dimensions or the more information we have in Z, the better it is, generally speaking, but it becomes trickier to visualize this entire Z stack. So we want to understand all of the data together, not just a single slice. So we either need to go slide back and forth on this thing to constantly consider what is actually happening in our Z dimension, or we can do some things with that which we will discuss now. So let's say I've got this Z stack here. I don't actually have this image, this I got from another website, but you can see I can look at all of the slices individually, and then I can get some understanding of this. But this is quite tedious and not very, it's cumbersome is the word I put here. So what we would typically do is we would summarize this information in one of many ways. So one way you can summarize it is just to do a simple Z projection. So you just open your Z stack. I'm going to make an example here. Let's just take this example. And I want to visualize this Z stack now. Instead of having to slide across like that, I want to visualize this as a single thing. Let me just duplicate this so that I don't destroy everything. Not the image, duplicate the stack. Duplicate the entire stack. I can now go to image, stacks, and then Z project. And then I choose the method of projection. So maximum intensity, average intensity, take the sum of all of the slices in Z, take the minimum intensity, standard deviation, median. All of these have different uses. Let's just take one of the most common ones is probably maximum intensity. And I'm now simplifying my image to just being on a single 2D plane. So I've squashed all of the information. I get a good understanding of the structure of the cell, but this is for visualization purposes, not analysis purposes. You don't want to throw any data away ever for analysis purposes. But to understand what is the structure of my cell, if I was to look at a single slice here, it's not, maybe not the best example, but if I was to go to a single slice, I don't know, on this front, I don't really have a full understanding of my cell. This slice communicates something, but not everything, and this projection communicates a whole lot more for actually what is the structure. So now I can maybe have them side by side and say, okay, well, I see this structure going on here, but oh, it's in the context of the larger cell. So a Z projection is a very helpful tool for that. Another type of projection is orthogonal slices projection, and it's not so helpful if the Z depth isn't very high, but this one is kind of sufficient to make the point. So if you go to image, stacks, and then orthogonal views, then you can, this one is quite low resolution in Z. I think I'm going to open up another cell here quickly. Okay, so I can see this one has 47 slices, so there's a little bit more resolution in Z. If I now go to, let's just make this a little bit smaller, image, and then stacks, orthogonal views, then I can more easily see now what's actually happening in the depth as I go to different positions. So as I drag this cursor around, I am now looking at it as a side view, essentially, cut through at that point. So where this line is, this is where I cut this thing through, where I'm looking at it. The horizontal line corresponds to this, but it's now showing me what's happening in depth as well. Okay, now another example of this might be if you go to open samples and then MRI stack. Usually MRI is viewed in this type of way. So I've got this stack of the brain. It's a fairly low resolution stack actually, but if I go to stacks, orthogonal views, here I can now either still go through the depth, or I can more easily see now, oh, if I was to, come on, it keeps resizing it, but if I was to move through the brain, you can now look at the, I don't know, they call it the sagittal and longitudinal slices, cortical slices of the brain, so that you have a more nuanced understanding maybe of, from that perspective, how it changes, even though your stack was maybe taken from the top. So it's a very helpful visualization tool, especially for MRIs. Because for an MRI, something like a maximum intensity projection doesn't really make a whole lot of sense. I'm actually kind of curious to see what will happen if I do Z project. Come on. First I have to get it back to that slice format. Open samples, MRI. There's the MRI. So image, stacks, Z project, maximum intensity. I get that, whatever you're supposed to make sense of that. But maybe there's some benefit. You can see the ear is on the head. I couldn't see that necessarily before. So yeah, but different ways of working with Z stacks. There's also a way to visualize it in 3D. So if you, instead of going Z project, you go 3D project, you can create these nice animations. It's basically still a Z stack that you can slide through, but it's now, every slice is now a different angle projection. There's also a way to visualize the volume altogether. So if I've got this thing, I'm not sure how well this will actually work now. Let's try with the MRI maybe. And I can go to plugins, and then at the bottom here, there's something called volume viewer. I actually had an issue with this yesterday as well. This usually works very well, but for some reason, Fiji just decided to bomb out yesterday. So the volume viewer isn't working, but then you can actually view the volume more together as a three-dimensional structure. There's another one called the other plugins 3D viewer. Let's just see if that one works now for me. Take the MRI stack, everything. Okay, so it's now pretty squashed, but you get the idea. I can look at it as a three-dimensional volume. You can. The way you would fix this is you would first need to define your properties. So on the image properties, you say what is the pixel, the voxel depth. So I don't know, let's just make it 10. Usually you would get this from your metadata as well. So the metadata would say what is the width and the height and the depth, the Z step essentially of your image, and then you can get the general scaling correct so that once you then do this volume viewer or 3D viewer thing, it should have the correct scaling to it. This is not great. I think it's scaled in the wrong direction. Looks like an alien. But yeah, you get the idea. That's how you would compensate for that type of thing. Okay, so I have already shown you the metadata, and this is just another kind of discussion of that. And I already showed you the scale bar, so I'm not going to repeat all of this, but there are more advanced scale bar tools, such as, I think it's a scale bar plus, which is a plugin you can download. So if scale bar is important for you, you can get more customizability than the out-of-the-box scale bar features that Fiji offers.
What needs to be in your metadata to just be able to put in a scale bar?
Yeah, so the pixel width, pixel height, and voxel depth, those things. So in other words, a single, I haven't explained this. If you've got a two-dimensional image, it's called pixels. If it's a Z stack, you call each individual element which contains intensities a voxel, a volume element. So that voxel needs to be defined in your metadata. What is its width, height, and depth in terms of the the way it was acquired. So yeah, and it usually says it per voxel. So that per voxel width, height, depth, and then everything else kind of follows from that.
So
Yes.
label it in microns. You have to get this.
You have to?
You have to get this plugin.
No. So as I was showing previously, I think I might have destroyed the metadata on this image. No, it's still there. So it still says, it knows it's 132 by 132 microns. So I can, in Fiji, by default, just go to Analyze, and then Tools, and then Scale Bar, and then we'll automatically add a scale bar. I can now say how wide it should be. Let's say make it 20 microns. You can already see it appearing at the bottom there. Thick it should be, I don't know, 10. What should the font size be? 20 or whatever. So I can already built in easily do all of these things, but if you want to have more control, there is a Scale Bar Plus plugin somewhere. But yeah, out of the box, it's still fine. Maybe just a quick tip here as well. In Fiji, there are so many features hidden in these menus, and it's hard to remember where they are. So there's a search box. I didn't know about the search box for the first year or so that I used Fiji. So in the search box, if you don't know where is scale bar, then you just type in scale, and then it will search for it. It's like, oh, okay, scale fit. Okay, well, I want to do a scale bar. Okay, great. Double click on that, and then it immediately runs the thing you're looking for. So that's really a time saver instead of digging through menus and trying to figure out where's the thing again that I'm looking for.
Is there like a specific site where you can get the plugins, like the Scale Bar Plus, or do you just
Yes.
Google Fiji Scale Bar Plus and click on the first thing that pops up?
Yeah, so there are multiple ways that you can install plugins. So first of all, you can create your own plugins if you can code in Java. But most plugins that are officially supported by Fiji, you can get by going to Help and then Update. So this is now within Fiji. You just click on Update. And looking if there are updates online somewhere for all of the things that are installed. It says, okay, everything's up to date. Then you click here on Manage Update Sites. If you click there, then you can usually just search for the plugin name. So Scale Bar, okay, that one's not actually here, but you can see there's a very long list of very commonly used plugins here. So I think later on I'm going to demonstrate this Stardust. So this is not installed by default, so you can just then click that Stardust, okay, great, apply and close, and then it will say, okay, great, now you can install that plugin. So that's probably the simplest, but you usually discover that by first Googling. So I can then say your Stardust Fiji, for example. And then I say, oh, cool, there's the link. It's on the Fiji website. I can get more information about this thing, and it shows me how to use it. But it doesn't actually allow me to download it here. Well, it says download, but then it just takes me to Fiji. It doesn't actually take me to the download of the plugin. So if the plugin appears on the Fiji website, the way you install it is that process I just showed you. You just search for it, it will appear in that list. The other way is, for example, there's this one we'll use later called PSF Generator. This is not a Fiji supported plugin, so you wouldn't find it in that list. So then you would just simply go to, you would hear about this plugin or discover it or whatever, and then you go to this website. It will have a .jar file that is a Java format for a single package essentially. And then in this case, I want the Fiji version, so I'm going to download this jar file. And it's busy downloading. And then I can go to Fiji and say plugins, install, and then I just need to find where it was downloaded. In this case, it was in the downloads folder. So there's the jar file, and then I just say open, and then it will take me to this instance of Fiji into its local plugins folder, because this is basically all that it's doing. It's just simply copying that jar file to the plugins folder of Fiji, and then if I save it there, I already have it installed. But then once that's done, you usually just need to restart Fiji, and then it appears in your drop-down menu. There, great, PSF Generator, now it exists. But we'll cover this again a little bit later when